So today I am in Berlin with Thomas Steger-Hartmann. Thomas led the investigational toxicology at Bayer, where his team focused on the safety assessment of new drug candidates. With more than 30 years of experience in preclinical and environmental toxicology, his work sits at the intersection of biology, data, and regulatory science. Over the course of his career, he has been deeply involved in advancing how we understand and predict drug safety, including the development of in vitro and in silico approaches and large-scale data integration across the pharmaceutical industry. He is the industry lead of the VICTOR Consortium, which aims to reduce animal use in toxicology by developing so-called virtual control groups using historical data, statistical methods, and artificial intelligence. At the core of his work is a simple but fundamental question. How do we ensure drug safety for humans while moving toward more predictive, data-driven, and less animal-dependent approaches to toxicology? Thomas, welcome to the show.
Thanks for the invite. I'm looking forward to this discussion.
Yeah, I mean, we've known each other for a number of years, and I always thought that it will be good to have you on the show and have this conversation. So I've been looking forward to it as well. So the way I start the show, it's always the same thing. I think it's we will go into the science, into the technology, into the thing that we do every day. But I always like to know a little bit about the history of our guest, and we never discuss that. So that's something that I will certainly discover about you today. So how was young Thomas?
I won't tell any secrets. I think let's start around the age of 18, go back into childhood. The point is, and this is probably a little bit surprising, I didn't know what to do after my high school degree, and I was more into languages, history. This is something I wanted to study, yeah. But I wasn't really sure. I was a little bit into Romanic languages, and then I realized that linguistics is not something I'm interested in. I did a year of general studies, and then it was essentially dissecting everything which I do not want to do. And suddenly I realized the only thing which survived was biology. And this was then the point of time where I decided to study biology. Again, not really knowing what precisely in biology I would be interested, but I started, started in the University of Freiburg, then a couple of almost a year in US University of Michigan. And then eventually ended up more or less by coincidence in the major of plant physiology. Yeah.
Okay, so that sounds quite different from what you're doing today. So today you're working in toxicology. So what drew you from plant physiology all the way to your career in toxicology?
Yeah, that's really an interesting question, and I still profit from plant physiology at Bayer because we have different divisions. Yeah. And when the toxicologists from crop science come, they very often are surprised that I understand what they are working on because I understand the mechanism. But this doesn't answer the question how the move was actually performed. It was the time of the forest dieback, if you remember that. You were probably a little bit too young, yeah?
A bit.
Yeah. And everybody was in plant physiology was looking into the reasons for forest dieback and oxidative stress. And so this was my master thesis, but already in an in vitro system with cell culture. So I was looking a little bit into ecophysiology, and then came the question, what do you do for a PhD work? And I got the interesting offer to switch into ecotoxicology, into a new topic, drugs in the environment. And it was actually the university hospital in Freiburg which brought up this topic, not solely, but they found out a very interesting, it's kind of a detective story. They found increased halogen levels in the sewage water, and nobody actually knew what that means. And halogens, then you think about organochlorine compounds, and everybody was scared. And then they traced it back to X-ray contrast agents, completely different toxicology, because we are talking about iodine compounds, but they showed up by the analytical methods. If you characterize them, you could dissect this, but then the question was, what does that mean? I mean, it's quite logic. It's administered to the people, to the patients, and then you excreted.
Yeah.
It ends up in the sewage, but what does it mean for the environment? Yeah, and this was then my PhD work. And once in a while, I'm giving lectures at the university to students. I always tell them, if you want to have a job, create a problem and tell the problem to industry, and then you will get hired. And this is, by and large, what happened to me. At that time, there was still sharing here in Berlin, and we were cooperating with them on this research, and they said, we anyway want to dig in deeper. And so I ended up, I started in ecotoxicology at Bayer. Now, it's still not toxicology, even though ecotoxicology is a part in the toxicology department or in the function. And then I started to look in degradation. And this is then the onset of my really deep love for data collection in order to come up with generalized ways of interpreting things like structure-activity relationship. We try to do that for biodegradation. It's rather complicated, but the data collection per se was already complicated. And this is something which intrigued me. I learned a lot from the colleagues in genetic toxicology, which were at that time starting to build up their own system with structure-activity relationship or expert systems. I was looking into this technology, and this actually then brought me into the field of mechanistic toxicology. So I was essentially the one who introduced these in silico tools at Schering at that time, now Bayer, and build up first these tools in the genetic toxicology department, and then enlarged this group into mechanistic toxicology, which later on evolved in investigative toxicology. So this was not a direct path, but looking back, I find it rather logic.
So at which point did you realize that how central it was? Toxicology was central in terms of its importance to drug discovery and development?
Quite honestly, from the very beginning, because the way you work in the pharmaceutical industry is completely different what I was used to in academics. Yeah. So the first thing I got in touch with was GLP, and good laboratory practice. And I was intrigued how much they invested for good laboratory practice. And at the very first weeks, you think, wow, what a bureaucratic stuff. But when you dig in deeper, and they did a lot of training for all newcomers to get them understand why they do this, And the reason why they do is quality assurance, And this illustrated how serious they take it. So, quite honestly, Thibaut, in your master thesis or PhD thesis, did you discard some results which weren't really fitting into your theory? Be honest.
No, it's interesting because I had this discussion once with a friend saying, so when you record data, so for instance, I've done a lot of enzyme characterization, and then you have this point that you don't like because it doesn't fit. So I remember having this discussion with a friend. I was telling him, what do you do with this point that obviously is wrong? And most people say, well, I just remove it. I'm like, well, I'm sure I've done it. And I agree with you. I think we've all done things when you look at it retrospectively, it's a little bit unethical from a scientific standpoint. I mean, science is science. I mean, the data you get, they are the data you get. So to a long answer to your short question, I can't remember a specific time, but I'm pretty sure I removed a couple of points that I didn't like on some curves.
I'm not saying that we are all angels at the pharmaceutical industry. We are sinners like everybody else in science. But the point is, we can be controlled. So the sin can be detected. The sins I committed in my master thesis and my PhD thesis, nobody would ever be able to detect.
Exactly.
I mean, I still have all the data of my PhD thesis, so I could do it. And if somebody would ask for that, I would hand over the data. And this is exactly the point. Having this data, being able to control it, understand how it was transformed, because this is also something, data tweaking, And this was all controlled. Why? Not because they had so much manpower to install bureaucracy, but to assure what they do is trackable for the purpose in order to prove we did the right thing. Even if we fail, we can prove that at the time we did it with the best intention and it is not flawed on purpose, yeah, by criminal act. Yeah. And this is what showed me how important that is, because quite honestly, you could interpret in a way that you say, at a university, who cares? It's a publication which is without relevance after 10 years, so who cares? Yeah? But in industry, it might eventually end up in a drug which is given to patients, or which might, in the field of ecotoxicology, have harm to the environment, cause harm to the environment, and then it's very, very important to be able to track it back. And I had later on in my career a couple of situations where I was involved in litigations, and there it's very important to be able to prove, guys, we can show you the data, yeah, and let's go through it, yeah. So this is actually the point where I realized how important safety assessment, preclinical safety assessment in drug development is. Yeah.
But going back to academia, I think the good habits, they start early. I mean, especially if you start to, you're discussing with students. It feels almost harmless, as I said, to remove like 1 point here, 1 point there, but it's about getting the right habits from the very beginning. And of course, I mean, no one likes to repeat 10 times the same experiment to get a clean curve, but sometimes that's what needs to be done. Has your view of the field in which you're working evolved over the last several decades?
Yeah. So when I started, I would term the level of involvement of toxicology being dominated by descriptive toxicology. Yeah, and I do not mean this in any bad way. It's not pejorative. It's just the biggest part were the animal experiments. They were done and then described. Yeah. But the mechanistic exploration was only rarely done. So the pathologists, they certainly started to interpret the results, what could be the mechanism causing a certain lesions they found in the tissue. But it was not done in a systematic way, both because we didn't have the resources, the technologies, and if we had a serious finding, then we discarded the drug candidate and went to the next one. Yeah, and over the years, and I was just at the beginning of this evolution. People realize it's a waste of resources. We are accruing all these data, and in addition, there is this dramatic evolution of technologies and new insight. We can look deeper to prevent the same mistakes in a row. Yeah, and this was the onset of mechanistic toxicology. I'm not saying that it started at that time. Particularly genetic toxicology was working on this since decades. And funny enough, just the other day, I walked over in the office of a new group head in genetic toxicology, and she had on her whiteboard this famous drawing of Ashby and Tennant. Are you familiar with that one? This superstructure where you have all these potential mutagenic structure in one superstructure. Yeah, and I more or less grew up with this picture. I also had it on my whiteboard, and I was intrigued that it's still important. Yeah, telling. Yeah, you collect the data and then you create knowledge out of it. So genetic toxicology, I would say, was really the spearhead influencing this mechanistic thinking then in all other fields of toxicology.
So moving on, one thing which is central to the work of in toxicology is the role of animal models. And there is, especially now, a lot of discussion on how well animal models translate to humans. And I remember years ago when we started interacting, when at that time I was working for Elsevier, you were working with some of my colleagues, with Matt Clark, which I'm sure will listen to this recording.
Hopefully.
And you had a very nice paper, like studying actually how well animal translate to human. So, can you tell us a bit about what does the evidence actually tells us about how animal finding translate to human?
Actually, you may be surprised. Not that bad as it is usually laid out in the lay press. Yeah? Animal studies are doing a good job for certain modalities. This may change with the new modalities. We'll probably come to that topic. But in general, they are not that bad as they are portrayed in the lay press. Yeah? And this is also one of the outcomes of this big data mining we did in Pharmapendum, and the idea actually came from Matthew, and I'm still grateful for this exercise because I could not have done it, A, without the data, and secondly, also not with my data science knowledge, because that's very small. The outcome is if we find a toxicology, a toxicity in the animal, we have a high probability to also translate it into humans or find it in the humans. And one, it's not a proof, but it's a certain evidence that animal studies are not doing that bad is think about how many serious failures we had in clinical phase 1 or 2, serious failures. What comes into your mind? And please do not start with thalidomide, because this is pre.
Okay, that's the one that I wanted to say. So now you're exposing my lack of knowledge on this specific topic. But tell me,
it's De Janeiro, which everybody knows, yeah. And then this case probably I can't recall. I should have looked it up in preparing myself to this conversation. It's the Bial case in France. Where they were working on a CNS drug.
Yeah.
And both cases are very peculiar, but in assessing the cases, one realized that given the knowledge we had in toxicology, everything was correctly done with the animal studies. De Janeiro later on resulted in a completely new in vitro assay, cytokine release, to test for this storm event they saw. Bial, I didn't recently dig into the outcome, but to the best of my knowledge, one could not have prevented it. And now I'm going back to thalidomide. The problem is in toxicology testing, we do not know what we do not know. It's a fundamental heuristic problem, and this was the case with thalidomide. Thalidomide led to the development of full developmental toxicology with a completely new set of assays. And now permanently reproaching animal studies that they do not predict, I think it's flawed. And again, this sporadic evidence that we don't have these serious cases in clinical phase 1 actually tells me that animal studies are not that bad. Now the people come like Bailey et al. And say, yeah, but the adverse events. Now think how that happens. An adverse event per se is not a direct, always happening toxicological effect, but there is a statistical thing behind it. Yeah, you have it 1 in 10, 1 in 100, 1 in 1,000, and sometimes only 1 in 100,000. How many animals do we have in a test? 60. If we talk about rodents, much less if we talk about the non-rodents. So purely from a statistical perspective, we can't detect it. So it's nothing against the animal study. One could now be cynical and say, okay, let's enlarge them. Use 10,000 rats. Not really sure. I could imagine that we increase the productivity.
But we will still have a problem of genetic homogeneity with animals.
Here we go. You have, you have the problem of genetic heterogeneity. You have predisposition issues, which you normally don't have in these outbreed species. Yeah, and therefore you can't reproach it to the overall model. It's individual bits and pieces which contribute to partly lacking productivity.
Yeah.
So overall, I would say it's not that bad as it is permanently portrayed. Yeah? I was recently discussing with somebody in the field of NUM where we were thinking, what would we need if you start from ab initio as a toxicologist, not even knowing that there are animal models, yeah? And you would say, okay, we have cells. We have in silico, yeah, but for in silico we need a training, yeah? We need the cells to train in silico, but cells, we need tissue to have the tissue interaction, the cell intracellular and intercellular interaction, but then we probably also need inter-organ interaction, yeah? Metabolism, sending back metabolites to end organs. But then we will probably also need a vascular system. And you probably have seen the movies from Uwe Marx and Tissues, very inspiring, where he has this lying human being, which is then dissected and the individual organs put on a chip. That's a little bit how you would think, yeah? And then you realize, okay, it's getting more and more complicated, more expensive. Sometimes we do not really have the technology at hand. For example, vascularization with endothelium or tissue in the chip. Still an issue. I also haven't really seen a functional kidney. Liver is fine, lung, partly. And eventually, after 2 or 3 days of discussion, one will probably come up and say, why don't we use a holistic model like an animal?
Yeah. I definitely want to come back to NAMs later on, because that's obviously something that everyone is discussing. But if we were, trying to summarize on animal models and simplify, so where is it that they work well, and where is it that clearly they fall short?
Yeah, we recently had a publication, Beilmann et al., Mario, my colleague and friend from Beringer, who coordinated this publication from pharmaceutical companies, where we dissected the question into modalities, and I was already alluding to that. There are a couple of modalities. For example, if you talk about humanized monoclonal antibody, then it's really the question whether a rat study... No, it's no longer the question. A rat study doesn't really make sense, yeah? And even a monkey study, particularly if you don't have the target you want to influence with a monoclonal antibody, then the question is really, is it meaningful? And couldn't you test this with in vitro systems, with organs on a chip or things like that? For similar might be with oligos, but we need a little bit more experience, particularly with the organs on a chip, whether they can detect effects where we know something should happen. With classical small molecules, I think the animal models are not doing that well, particularly if we enhance them, meaning that we should not blindly use rat and dog. Nobody does it anymore. For the rats, yes, but for the second species, there is always the discussion, what is the best species? And now you could even increase this discussion insofar as you say you have a target safety assessment before you start the animal studies, and you look into what organs might be affected. Do we have the same genetic constitution or expression profiles in these organs between human and the rat? If not, what are the differences? Yeah, and the data is all out, and you can dig in deeper, and then, for example, enhance certain animal studies that you say, okay, normally gene expression is not a routine parameter. You could include it in order to see, is an effect now predictive for humans? Is it correlated with gene expression for targets where we know they are there, or are they related to a target, an off-target which is non-existent in human? Then we know that's not predictive. So enhancing these studies, and then you could do it more target-oriented.
So if we understand some of those limitations, so what is the reason why we still rely heavily on continuing to do some of those studies like we've done before? So is it a matter of science, regulation, inertia?
All the three. You named it.
Okay.
You named it. Before, I would have come up with the same three categories. Yeah. And I do not really know which is the most important. I would say, in terms of quotation in industry, it's probably regulatory. But I always tell people this is intellectual laziness, because the people in regulatory authorities, 80% are scientists, trained scientists, and good trained scientists, meaning they think the same way. The objectives are slightly different because they are responsible to protect the patients. From a regulatory legal point, we are also expected to protect patients and the environment, but we are here to find new drugs and eventually we have to market them. So there is a certain conflict of interest, but the basis of science is the same. What I want to say, if we permanently target and say, yeah, we would use it, but there is a lack in regulatory acceptance, I would say this is intellectual laziness. Because you can start the discussion, you can even go for a qualification and ask EMA and FDA, they have their dedicated programs for that, and ask them, is this acceptable? Yeah, and we did it in VICTOR, we probably come to that. It's possible. So this is a reason which is often quoted, but probably the least important, particularly nowadays. In the past, it was a cumbersome process to go through validation. But nowadays, they are much more open and agile, I would say. Inertia, yes, we have done it always like this, yeah, and in combination with guidelines or guidance and SOP. I frequently call this a bumpy transition road. On the one hand, these guidelines are good, and by the way, they reduced the use of animals because you have a mutual acceptance. When I started, ICH was just in the beginning, the International Conference on Harmonization, Council of Harmonization. Former times it was had a different name. And at that time, we did a reproductive study for Japan, which was different from the one we did. And people were already saying, this is for Japan. Yeah, they want it like this. So we had a tremendous cut in animal studies already with ICH. And the same holds true for OECD, because if you have an OECD guideline, the implication is mutual acceptance. They can't come back to you and say, we want this and that. Yeah, if you have done it according to OECD, interpretation is a different thing. So it's not bad, but it's carving the whole thing in stone. Yeah? It's making changes slower, and very often people say, yeah, we can't do it, it's written in the guidelines, and then you combine inertia with regulatory. Yeah? Science, probably the least. Yeah. Because just recently with AI and advances in data science, I'm absolutely amazed how quickly you can now accrue data and also curate data. When I was talking about the genetic toxicology Ames test data we were collecting to improve the in silico system, we were thinking about databases, access or things like that, completely user-unfriendly. Now you have it at a fingertip, a whole 4-week toxicity study. Yeah? So that's more technology, not so much science. But that also allows us to put the different parts of science together quicker. Yeah? And therefore, science is probably the least. Inertia, I would say, almost the biggest.
You worked on the so talking about technology and talking how technology can impact this type of work. You worked on the concept of virtual control groups. Can you explain for those who don't know what are virtual control groups? Can you explain what it is?
Yeah, that's so simple that I'm always struck that nobody else has come up with this idea. I mean, as said, we do the animal studies in a very standardized, harmonized ways according to guidance. As a consequence, and it's always set up in a pretty much similar way. You have a control group, a low dose, mid dose, high dose, and this goes back to Paracelsus. Yeah, if I always say, if I give a lecture on toxicology, if you have understood Paracelsus, then you have understood 80% in toxicology.
So the dose is the poison.
Exactly, you got it.
Yeah.
Now, what is the control? The control gets the same, we call it vehicle. So the basis of the formulation of the test item. Test item is the formal description of the compound, which is tested formal according to GLP. Test item is the correct description according to GLP. And the test item is formulated so that you get it into the animal either via oral gavage, via food, via intravenous injection, and depending on what route of administration you select the vehicle. And then the control just gets the vehicle without the test item.
Yeah.
And this is very often the same thing. Yeah. So the age of the animal is the same. Route of administration is the same. Housing is the same. Temperature, light, day cycle. Why do we use in each and every rat study 12 animals, 6 females, 6 males, which these things the fellows have seen a couple of weeks before? Yeah? And we said, let's use the data and compare and save 25% of the animals in the study. You have to put that data in a repository. You have to create it, and then you have to develop standardized terminologies, because the one may call it transaminase A, the next one says, no, it's called differently, and then you don't find it. So the basic things in data curation, data science, you have to overcome this, but as soon as you have it, you can also compare your 3 treatment groups to digital twins, I would call them, yeah, and see whether the results are the same.
So why don't we do it all the time?
Because the way these studies are described is laid out in OECD guidelines. So if you deviate from these OECD guidelines, you'll use your mutual acceptance. And that's the simple reason. So people now say, no, you're contradicting yourself because this is regulatory. Yeah, it is partly regulatory for good reason. And I would not recommend immediately discarding it. We now need to show that we get the same results with the virtual control group comparing to the treatment groups as we normally do with the concurrent. And as long as we haven't shown that, we can't use it. And I would not recommend using it, because then we risk to impair the safety of patients. And that's the ultimate goal why we are doing it. Yeah? If I lose certain findings, or I could also in theory come up with new findings which could kill a valuable drug, then I have a problem. So I need to show that I'm replicating the original study with the virtual control group. And this is what we are doing in this project.
So how far are we from actually being able to have this as a standard in the industry? So you say we are doing this project, so we are in the process of doing it. So when is it, in your opinion, that we will have this as a normal procedure, as opposed to something that is still new and where you need to convince people? I remember also, when I was working at Charles River, this was still something new, virtual control group, and not every client was open to that.
Yeah, for the simple reason, because you can do it in those range finding studies, because then there is less regulatory, less regulatory framework. But to answer your question, not so long in time, I would say, because on the 31st of March, EMEA has published a first qualification opinion on the use of virtual control group in dose range finding studies. Yeah.
Okay.
But we also only applied for those range finding studies because we wanted to get the foot in the door and test the water. How would they react? How is such a qualification procedure? We it's a qualification procedure and not a validation procedure. We can discuss the difference. Yeah. It's quite an interesting difference because animal studies have never been qualified and never been validated. That's an interesting thing, yeah. So we applied for a qualification for dose range finding studies. We intended to have it for all species, but the data we submitted to EMEA was not sufficient for all species, so they said for rats. But the speed they worked on that was surprising. And this actually brings me to this point. They are scientists. And they are open for this scientific discussion, even though we have partly different objectives. Yeah? But it's very important to start this discussion and not just start a finger pointing. We would do it if they would allow it. Yeah? Yeah.
I think it's important what you're saying, that the regulator is not the enemy. You know, the regulator exactly. is the partner. And I remember also having actually a partnership with the FDA, where they were coming to the industry to ask for certain data because we were working on a project, and we'll come to that when we talk more about the AI role in toxicology, but one of the projects was to try to assess hepatotoxicity, to predict hepatotoxicity. So I remember my contact at the FDA came to us and say, we don't have data set on hepatotoxic compounds. Why? Because the industry never comes to us with something that is hepatotoxic. So everything they had was the positive, the compound that didn't trigger hepatotoxicity. And of course, you can't train a model if you don't have the other side. And they say, well, dear industry members, can you give us some of the compound that you had where you recorded the hepatotoxicity? So, and the point that I wanted to make is, they are actually, the regulators are really open to new methods. They are open to working with the industry. So it's really about collaboration with the regulators, as opposed to just being regulated, because it's just, not just actually, it's another perspective on the same work actually.
And as said, this is also my experience with this qualification, which is still ongoing, because what they published was a draft qualification opinion, and it's now in public consultation. But the whole experience was extremely positive. They were providing very constructive feedback. It was not easy going, yeah. But also the concomitant press release of EMEA, where they said it's the first qualification of a NUM ever for preclinical purposes, EMEA did, made us a bit proud, I have to say. And they also said that it is a blueprint. What else can you get? I mean, they are handing over the map, the roadmap. Please continue. Yeah. Here is the direction. Yeah.
So you're the industry lead for the VICTOR Consortium. So can you tell us more about the initiative? What are the problems you're trying to solve, and at which system level it is?
Yeah, important. We founded this consortium because, and it came out of a previous consortium, namely eTransafe, where we got to know each other. eTransafe was a consortium where we actually collected preclinical safety data. So in the light of this delete thing you mentioned, because we also said we have all this wealth of data in the archives of the pharmaceutical companies. Now we need to leverage it. We need to make assets out of it, data as an asset. And some companies were not providing the full study data because they said treatment groups and compound information is proprietary, but we send you the control data. And first I was really a little bit upset, and what should we do with the control data? And then I had a discussion with a couple of people, and they said, yeah, but if we show that the control data is stable for certain parameters over a period of time, we could not only use it as a reference value for discussing historical control data, but use subsets for a specific study and replace the concurrent controls. And this was the birth of the idea, which was already published 2020, so quite a while ago. But then a couple of companies continued and said, this is fantastic, let's try this. Yeah. And we first had a little collision of the welding and had first contacts with EMEA shortly after eTransafe, and then said we need another consortium because you can't do it alone. A single pharma company can't do that. Also because the fees for such a qualification are much higher than you do it with a consortium. That's a side aspect, but also in terms of credibility. Yeah. If you donate the data from several companies, if several companies look in the same processes, so you come and develop such processes, then it's much more credible as if you would do it on a single company basis. And then we applied for another IHI project, and this is actually VICTOR. So we said we need a common database. We need a controlled terminology, which is partly out with send C-disk, you're probably familiar with. However, there is such a lot of creativity to implement send in the different companies, so you can't easily use the Merck data to compare it with the Bayer data. So this is a tremendous effort. Then you have to come up with a procedure. How do I compare the virtual control group with the concurrent control? And this is a big statistical question, because at the end of the day, it comes down to the question, how similar are animal population, with what tools do you prove or show evidence? You can't really prove it. Yeah? Show evidence that the two populations are similar. You have the animals which are bought for the original study, some 6 months ago, and this is subpopulation. You use your VCG groups, which is also subpopulation. How do you show that they are actually one of the same population? This is a statistical question, and we have really brilliant and dedicated statistics which look at this, and this is without any precedent, because who has done this before, really showing the population have the same distribution, belong to the same, maybe with a slightly different variability, but what is then still acceptable? And these are the problems we try to solve in VICTOR.
How did you manage to get the different companies, especially the lawyers, to accept to share data? I'm asking because I worked on similar projects, and actually at one point in my career, one of my jobs was to negotiate data sharing agreement. I spent more times with lawyers than I ever wanted to, and reading contracts, which was not fun. Most of the time, the response when I was asking people to share data, even in non-competitive ways, was no. I think now things are evolving because people understand that to train model, you actually need to have enough data, but it's a very slow process. Yeah. And I still remember a lengthy discussion where people were just not willing to share data.
I shared the same experience, Thibaut. When we started with the first project, eTox, which I conceived with my old friend François Pognon, now retired from Novartis. You also know him.
I know him, yes.
The project, I can't exactly recall when we started, but the first 2 years after the start of the project, the computer scientists, that's the way we called them at that time. Now you would then probably call them data scientists. They were just scratching. When do we get the data? And we were still talking to the lawyers. Yeah. And the toxicologists said, okay, we understand what you want to do, but we don't have the time to convince the lawyers. Yeah. And so we had to establish internal processes for clearance. Yeah? And at Bayer, I would say at that time we were quite efficient because we identified then 3 levels: IP department, litigation department, and also internal preclinical development. And we always sent them a batch of compounds which we wanted to release, and then they were checking, are they under litigation? Whenever under litigation, no sharing. Then IP, patent protected, okay, no longer a problem. And is there any special interest from medchem or so, because it's a special cluster of chemicals? And if all was positive, then we could share it. But it took us more than 2 years to build up such internal infrastructure. And Some companies weren't able to do so, and at the end of e-tron Safe, the follow-up project, some companies still couldn't provide data, and therefore only provided control group. Now, the big thing with control group, no compound information, except the vehicle, but this is trivial, and no structural information. So there is also the effects of treatment groups are not shared, so there is no IP.
I worked on some of those data sharing project where we were actually asking to share compounds, and that's triggered all sort of headache. So if the VICTOR project succeeds, so what is it that will concretely change in how toxicology studies are done?
One simple thing is that you could save around 25% of the animals of an individual study. I'm not saying overall, because once in a while you need to renew the data in the database. Then you have specific studies, and funny enough, we are doing this now with a pharmaceutical perspective, but it's probably more relevant for the chemical industry, for the REACH studies which are done, because they are even more standardized. We try to build in a lot of parameters even in the regulatory studies. So like, for example, ECG or micronucleus. And then it gets difficult to find the matching animals in the virtual control group, because time points might differ, or when you do the ECGs, when is Cmax and things like that, then this will make the thing difficult. And I've been asked frequently, but then how many of these studies? And I don't come up with an estimate, because then I will be put under pressure. And 25% of an individual study where you find the animals. That's one thing. We have recently allured the interest for our database from EFSA and ECHA. Even though they say virtual control group, do it first with EMEA and then we'll have a look. But this tremendous repository of historical control group, can we access it? And sure, we will discuss how, but the interest in a well-curated and easily accessible historical control data is a value per se, independent of VCG, because then you can look for rare incidences. You go in literature, there are alternative databases like, for example, the carcinogenicity database at Rita Fraunhofer in Hanover. But we probably have much more animals, much more data, and then you have a better understanding also what influences certain parameters, like correlations. And therefore, drilling in now into this data with AI and dedicated statistical procedure will provide a much better insight into the studies we are doing and what are background incidences. I give you an example, and this is probably already bridging to AI. One of our partners is Barcelona Supercomputing. And it's a kind of a funny thing, because one brilliant lady was coming to me and say, if I use your data, I can predict the sex of your animals. And I was like, oh, wow. You're looking into ovary and testes or what? No, it's different. It's different. So they take the animals and subtract the sex-specific information which is in the database. Then they calculate that. They look in the spontaneous incidences, the background incidences. Then they correlate these background incidences with specific gene expression profile from the literature. We don't have that in the VICTOR database. And then they realize that there is a clustering of specific background incidences. And when she explained it to me, I was like, wow, more of this. Because then if we do a study and see, even in the treatment groups, a specific organ toxicity, and go back and understand that this has already a predisposition for male and female, then we have a much better understanding. And you couldn't do that with the data collection you have out in the literature. And now we are still joking, because when he first approached me, I was like, you know this joke of the business consultant which tells a shepherd how many sheep he has? You know that joke?
I think I know several versions of that joke. I mean.
And I was like, but I know what the data of a female and a male rat, but then suddenly it made a click and I understood what she's doing. This is really dramatic.
So you mentioned AI, and we spent almost 40 minutes talking without talking too much about AI, which I think is a great achievement. But let's talk a little bit about AI in toxicology. So, as I said, everyone is talking about that. Every CEO in the pharma industry has to do AI. But if we talk about specifically AI in toxicology today, what is real? Is there something that already makes a difference, or are we still just at the very beginning?
No, it makes a tremendous difference. Yeah. But not that we now start to predict toxicities with AI.
So you can start from a compound and say this compound will have hepatotoxicity or cardiotoxicity or something.
This is very imaginary, and I think we are still far away from that, even though people say we have all the tools. But the effects of AI are much more mundane, in a way that it speeds up work dramatically. Now let's look at it from different angles. Nobody talks about databases anymore because you get the data together with different systems. At the end of the eTransafe project, we had so much learning out of the eTransafe project that we set up our internal Bayer preclinical information center, so a repository, and it was built up like a database, and it's really fantastic, and many companies now envy us for this. But then I had a brilliant colleague who said, databases or setting it up in a way of a database, that was the past. Now we are using prompts. And I was like, And now we have a system where you type in, and I have always dreamed of a thing like that. I type in, show me all the compounds which cause liver toxicity in rats, indicated by a combination of AST and ALT elevation, which did not occur in dogs. And you get the results, and you have it within seconds.
And you trust it.
I trust it because, and you interrupted me, but this
Sorry.
is no, that's absolutely okay, because this is a fundamental question. I trust them simply for the fact that the references with the original reports is there.
Okay.
And then my brilliant colleague has linked this to the original platform they built, where you go into the data and visualize it. So you have, on the one hand, the PDF report, you have the raw data, the sent raw data, retrieved by the prompt, or you first retrieve the prompt, then you get the report as reference, you open the report, and then you can go back to the database and you can visualize the data. And this probably takes you 10, 15 minutes. This was a master thesis 5 years ago. Yeah. And now imagine you get a question from during a submission, and this is where also the elder generation, I belong to them, of study directors and also project toxicologists were convinced when they had to answer a question, can you provide similar compounds, or can you provide detailed information on this and that? And they were sitting in the archives for a day and going through reports, highlighting, typing, pasting and copying. Now you do it in 10 minutes.
So you're describing, so what you're describing is, again, it's a very important use case. It's finding existing information, which, as you described, used to take a lot of time because the information was all over the place, and retrieving it was challenging. And this is where AI and the tools that we are using, the LLM-based tools, are doing a fantastic job. However, what we were saying at the beginning, predicting toxicology is still science fiction at this timing point, or are we making progress?
Absolutely. Another example is target safety assessment.
Okay.
Yeah. And it leads us a little bit back to a question you asked in the beginning: how did toxicology change? When I started out, it was a kind of a sequential procedure. Yeah, you had first the pharmacologists coming up with a new target, and then an animal model testing it, and then it went to PK testing exposure. Does it get into the into the body? And then into toxicology. And now it's much more iterative, yeah? Meaning we are with them in the very beginning. Target safety assessment, this is not entirely new, but in the past we send out the question to a CRO, like your former company, yeah? And then they came up with a literature search and did the target safety assessment. Now the target assessment, target safety assessment is done after the first idea of a new target, yeah, including all the information you have in PubMed, but importantly also the internal information. One dramatic advantage of AI is that you do not have to do the manual creation. I mean, you have to have a good understanding, and I'm not saying that hallucination is not a problem. It is a tremendous problem. But if you can then go back and check it with your internal data, you can overcome this issue pretty fast.
So one thing we hear a lot, in the past few years is NAMs, so the new approach methodologies. How do you define them in practice?
I use the quote of EMEA. I like this question. I like this question because many people say, num. Yeah. Have you heard that? It sounds
No,
like dog food or
Yeah.
so. Non-animal method, new approach methodology,
Yeah.
which I think is semantics. And there was recently a paper, I can't recall how they abbreviated it, but it's made a pun out of it. I can't recall it. Anyway, usually NUM is an in vitro or silico replacement. Interestingly, EMEA said it's a new approach, also including, I'm not literally quoting them now because I can't recall the literal quote, but they are saying a modified in vivo approach would also qualify for NUM. And they had their 3R working group stakeholder meetings where they presented this definition, and I was secretly applauding because we consider VCGs as a NUM. Yeah. But then often enter into the discussion with the in vitro and silico toxicologists. This is not a NUM. Yeah? I think this is a ridiculous discussion because it's a question what the overarching goal is. And my overarching goal is advancing science and improving ethics, if possible. Yeah? And therefore, my NUM definition would also include new approach methodologies, including in vivo studies or animals. Yeah.
Okay. So what are you mentioned VCG that we talked about extensively. What are some of the approaches that you think are gaining traction today that you would put in the NAMs category?
What I'm particularly fond of is organs on a chip, microphysiological systems, and this was a couple of years ago still something I was active, or we were active. I find this model extremely fascinating. We have worked on particular combination of organs, which were quite striking because we were able to show that there is a replication of species difference on the chip. Yeah. So we were combining liver and thyroid, human and liver and thyroid rat, and we were able to show the differences which we know in the literature, well reported between interaction between thyroid and liver in humans and rats. We were able to show that, and this was absolutely fascinating because for a couple of reasons. First of all, you show an interaction between organs, tissues in vitro. Yeah. Secondly, it was an interesting learning exercise because when we started, the biologist or the medical-driven toxicologist always said this is not possible because if you want to have a thyroid, you need the whole axis. Yeah. And so you essentially need 2 or 3 endocrine organs which are steering, controlling each other, and then combine it with the liver. And I was like, yeah, I understand that. And then when we entered the discussion with the engineers, with the tissue engineers, they said, scrap, we just pipette TSH in the rhythm as it would be biology. And this is ridiculous, but I was so struck by this thinking of Occam's razor. Are you familiar with this expression?
Yes.
I really liked it. And I came across an article where somebody wrote why a biologist can't reconstruct a radio. We are thinking sometimes too complex. And I think if you have a good definition, what do you want to do? Call it context of use, call it fit for purpose, then these systems contribute a lot. Yeah? And they are in use, particularly in a tiered use. Yeah? Before we do the animal studies, we apply them and sort out the bad guys, yeah? And also come to a better assessment of species specificity. Yeah? In silico is also making rapid progress because we are now much better to integrate different information. So what I envisage is in silico prediction based on agentic AI, yeah? Where you say, okay, predict me the liver, the DILI. Yeah? And there are systems which are already quite good. Genetic toxicology, that's a given already. And then you can combine it with PK prediction systems, yeah, to talk about exposure. When do we reach the DILI threshold? Yeah, and I could imagine that you combine more such systems to eventually build such a digital twin, also from a toxicological perspective. I think this.
So there are companies working on on virtual cells or even virtual organs, and I remember writing about that, about some of the experiment, and this is actually the first time when I wrote about that and posted something on LinkedIn where I was actually insulted by a few people saying, how can you post about stupid things like that? We are unable to do this. And I thought it was really interesting actually to get such a strong reaction from some scientists. Because I thought that it's kind of struck a nerve where, I think it's this contrast between folks who will think, okay, the only truth is the experimentation in the lab on in vivo or in vitro system, and we are nowhere near creating a digital representation. And the way I was argumenting was, well, it's a model. And by definition, a model has gaps. But then when you use a model and you simulate something and then you compare the result of a simulation with the reality, then you see where are the gaps and then you can improve the model. And that's what physicists have done for years and have made very interesting prediction. But I was really interested to see such a strong reaction from biologists. Yeah. So what do you think is what will it take for this kind of approaches to get broader adoption?
I think the reaction is probably triggered by a combination of fear and misunderstanding of a vision. Yeah? Particularly in the field of animal replacement, there are extremely high hopes and people driving this hope and promising that we can do it readily without. And this easily falls on public grounds, and there is this European motion saying we should do animal-free research, and particularly in Berlin we had also local motion. And such arguments, we can easily do it, fall on fertile grounds. Yeah? And this then struck the people who have the responsibility to drive the science and take the responsibility for the results. They say, this is not, he's one of these silly people telling that tomorrow we don't need that anymore. The point is you're expressing a technology from a visionary perspective. This is something valuable we could use precisely as you describe it. without any constructive model, we would not have come up with this chip technology of the two organs, because you need to understand your process in order to apply the Occam's razor. If you don't understand it, if you don't have a model, like a blueprint, like somebody building a radio, he has a blueprint, she has a blueprint, and then builds the radio. Yeah? And you build your model, and then you know what you have to measure in order to verify or falsify, in proper sense, only falsify. And then you correct your model, and that's the true value. So you have to express it in a visionary way so that people are allured, but some people interpret it as an overstatement, and this is caused by these promises which are out. Tomorrow we can take it. Yeah, we can do it without.
So yeah, you mentioned something. You know, I remember when the FDA published their position paper saying, you know, we can do without animal experimentation. So I don't remember exactly what was.
They didn't do it this way. This is interesting what you're saying, Thibaut, because it was then quoted and rephrased and rephrased and rephrased.
Yes, you're right. But I think that's precisely the way it was received and then, you know, disseminated everywhere. But I remember at that time, you know, I was actually not working anymore for, you know, as you mentioned, I worked for Charles River, which, you know, is a leading company doing preclinical experimentation. So obviously when the FDA published that, it was the day it was published, this paper, the stock price of Charles River and other company doing something went down, but the stock price of company doing in silico model went up 40%. And I think, to your point, it played on the false hope and promises of things that it will happen from one day to the other. The stop
premature hopes. I would not say false hopes, but
premature hope. So then that kind of lead me to, you know, the regulatory perspective and the FDA push, and in many ways also the EMA. I mean, the EMA might have a more subtle way of doing it, obviously. But what's your perspective from the regulatory agency and how they are looking at driving animal reduction?
I mean, I already talked about the European motion, Europe animal free. I can't recall when this motion actually was handed into the European Commission, but this actually then triggered the European roadmap, the European Commission roadmap. And it's difficult to talk about a roadmap because a roadmap tells you where you start and where you end, but they don't really know where they end. And I would be a little bit worried if we run into a situation we had with the ban of animal studies for cosmetics industry, if this is now extended to pharmaceuticals. And I think this is precisely the reason why they do not exactly know where the road will end. On the other hand, that there is this public pressure. Yeah? Yes. And this is absolutely okay. We are living in democratic countries, so it's not a single man's or women's perspective or decision. It's the desire of the population, the citizens, to reduce animal studies or eventually even become animal free. So the authorities have to deal with that. And I think they are now looking for fields where it is possible to show, we are qualifying things and we are dealing with that. We are not just ignoring it, but they also know, and this is again the point, they are scientists. They would also suffer from a situation where we are no longer allowed to use animals. And if you talk to people in the cosmetics industry, I mean, clearly enough, a trace test was a meaningless test, but the complete ban now has consequences that certain compounds can't be used for certain cosmetic purposes because you can't test them. Now you can start a discussion, is this good or not? For cosmetics, probably I'm not using a lot, and I'm referring to the ones which have been tested before. But if that field is extended to the pharmaceuticals with a fixed ban, that would scare me.
So I will ask you now an impossible question, as we are coming close to the end of our discussion. So if we fast forward 5-10 years from now, what will toxicology look like?
Yeah. As a toxicologist, being involved with regulatory interaction, I tend to be rather conservative and not that visionary. So that's a trait that's almost in the DNA of.
That's mandatory in the job.
Almost a mandatory, I would say. Yeah, you could also call it a déformation professionnelle. Yeah? But we have already touched on a couple of things. Yes. AI will play a better role, and we will, in parallel, also improve our understanding where the limitations are. I mean, hallucination is sometimes really funny. Yeah. How many publications would I have if I use the ones which AI creates immediately? But this is easily detected. Yes. So this will improve, and we will get better. And this also applies then to scientific hallucinations. So that is one thing. Another topic we only briefly touched on, new modalities. And the new modalities will require a completely different approach. Yeah? Oligos, humanized antibodies, and new targets, undruggable targets, will require completely new approaches where we also work closer together with the pharmacologists, which we already do. Yeah. So where will we be in 10 years? Probably more personalized medicine. And this would then result also in completely different safety assessment strategies. But also endeavor into fields where the things we do not know might even be bigger here. Because with small molecules, you can go theoretically through the off targets. Yeah? But with oligos, there is still a lack of knowledge. Yeah? And I could imagine that we bridge this gap. We will certainly include more in silico. We will include more organs on a chip, and I could even imagine that the technology around organ on the chip, how you prepare them, how you cultivate or create the tissue, will play a role. Yeah, that's my limited vision. But toxicology will still exist.
Well, Thomas, I think that's a great way of concluding our discussion. Thank you so much. That was fascinating, and I think we could go on for a long time. But I think also we are trying to have this discussion to have it in an hour, which is almost impossible because there are so many topics. Yeah. So perhaps it will be another episode.
Maybe.
Thanks again, and
it was a pleasure, Thibaut.